Good morning, everyone, and welcome to this webinar. In this webinar, we want to cover a few things. First of all, we'll talk about some of the upcoming OmicsLogic online and on-site training programs in bioinformatics. I do want to let you know that this webinar is going to be recorded, and so we will be sharing the recording of the webinar with all the participants and those interested to join one of the programs that we will discuss in today's webinar. We'll also talk about some of the online resources that we have prepared for self-guided learning. If you're interested in taking courses on your own timeline and schedule, we'll talk about some of those updated materials. And we'll also talk about some of the bioinformatics resources. So let's jump in. I will make pauses for any questions. There will actually be a time slot for you to ask any questions, but at any point in time, if you have something that you would like to ask, you can put it on chat. The chat box is open if you go to the Zoom bar, and in the Zoom bar, there's a three-dot button that says More, and you can click on it and open the chat. So if you have any questions during the session, please take a look there. If you want to have this webinar recording at the end and you have registered for this webinar, you will receive the recording. And you'll be able to also view that recording on YouTube and other social media where we will post it after the recording is processed and shared. So in this webinar, again, like I said, we will speak about some of the upcoming training programs, resources for self-guided learning, and bioinformatics research. And our goal is to introduce the topics and program details and answer any questions you might have about registration, about program participation, any topic questions that you have about any of the programs that we will be having, or anything else that you feel that is not clear and you would like that to be clarified. My name is Ilya Brodsky. I work for Pine Biotech, and I'm going to be sharing with you some of these details, but also online we have some people available that are from different regions where these programs are happening. So if you have a specific question about programs in India, programs in Nigeria, or programs here in the US, we can help answer those questions as well. So we have several upcoming programs that we want to go over today. There's a program that will be starting in, two programs that will be starting in March, and one program that will be starting towards the beginning of May. The first program that we'll talk about is Precision Oncology, Bioinformatics for Precision Oncology. Another program, Introduction to Big Data in Life Sciences. And we will have a program focused on bioinformatics for infectious diseases. So I'll go over some of the topics that we will have, that we will cover in these programs, and also talk about some of the general things that we offer either online, on site at different locations, or some of the bioinformatics tools that we will utilize. All of our OmicsLogic programs are designed to provide theoretical and practical training for those interested in next generation sequencing data and other high throughput data that talks about the molecular changes that could be studied using data analysis techniques. As you know, this data is becoming widely accessible and collected, focused on a variety of topics. And so in each program that we will discuss now, we will learn about the different types of data, how to process and prepare this data for analysis, as well as how to utilize data science skills to mine and visualize large data sets, and also present your research to peers and experts for feedback. And we will also discuss some of the opportunities where you might be able to interact with experts in specific fields and domains and be able to learn from them and learn about the data that is available. So the first program we'll discuss today is the Bioinformatics for Precision Oncology program that will start on March 9th, 2020. In this program, we will present the background of cancer research that focuses on oncogenes and tumor suppressor genes and different changes in the DNA and RNA, as well as the regulation of these genes at the genomic and transcriptomics levels. The registration for this program is already open now, and we have limited places if you want to join this program. But to join the program, you can follow this link, which is edu.tbioinfo.com Precision Oncology 2020. And thank you, Beepsha, for posting that link right there in the chat. You will submit a registration form on this link, and after your form is submitted, you will get an email notification where you receive additional information on the program timeline, topics that are covered, registration link, and other things as well. But what we'll do now is we will go through some of these topics and some of this information briefly, and then if you have any questions, you can feel free to either ask them in chat as I go through the topics, or at the end we will have a slide to pause and have a discussion if anyone has any questions. So precision medicine, as you know, is changing the way that we understand, diagnose, and treat cancer. This transformation is driven primarily by high throughput molecular data from patients, animal models, and large-scale cell line experiments. So in this program, what we want to do is explore how the various omics data types can be analyzed to understand the basic biology associated with cancer onset, development, as well as outcomes, and see how large-scale data collected from research projects as well as clinical trials provides an opportunity to better understand this condition and improve the precision of clinical oncology. So the program will offer access to expert speakers that will share their insights into various aspects of cancer research, cancer biology, the different data, and data analysis skills. And they will offer guided training sessions where we will use the different analytical tools, explore data sets, and learn about the biology that we can see by analyzing this data. In practical sessions, we will also together process, analyze, and learn how to interpret data and where to find that data in public repositories. We will break down the complexities of tumor development and the role of tumor microenvironment. So we'll talk about changes at the subcellular level, as well as data that is available from whole organisms, tissues, and combination of tissues like the tumor and the tumor microenvironment. We'll also talk about the DNA, genes, and RNA transcription, and how these elements can be studied using next-generation sequencing. So we'll talk about how the process of sequencing works, and what kind of information does it provide that we can analyze, and then we'll talk about how the data could be processed and studied. We'll also talk about some of the approaches in basic and translational research in oncology, where patient-derived cell lines, xenograft models, where tumors are grafted into an animal like a mouse, as well as clinical trial data, offers a rich collection of data to inform diagnostic approaches and development of new treatment of cancers, either based on new target pathways or based on existing drugs that might be repurposed to treat specific processes. As you know, the recent growth in high throughput sequencing of cancer genomes and transcriptomes has produced a big data problem that precludes many cancer biologists and oncologists from gleaning knowledge from these data regarding the nature of malignant processes and the relationship between tumor genomic profiles, transcriptomic profiles, and treatment response. And so we will learn about the different data types and how to efficiently process the big data that is available so that we can get some meaningful insights from that data. Together, we will map, visualize, and detect significant changes in the DNA sequences using reference genomes and paired tumor normal samples, where we will learn how to discover mutations, whether germline or somatic mutations in oncogenes, and as well as genes that suppress the oncogene function. We will study the expression of activated genes and pathways learn about methods to perform complete analysis, starting from processing to exploration of data to interpretation that leverages online databases of genes, pathways, and functional processes that are associated with found genes and functions. The found genes that we will learn about play a role in various regulation processes associated with cancer risk, tumor development, and other key factors that are typical for various types of cancers. And so the activated pathways that you see here on the screen, we will learn how to interpret them and how to analyze the changes in those pathways that are associated with different types of phenotypic information that we have about the samples. But at the same time, we will learn about the biology of tumor development and how the tumor and the signaling processes are dysregulated in different cancers because of the genomic and transcriptomic changes that we can study. We will also learn about some of the public domain repositories where data can be found, and lots of data sets on different types of cancers provide us with good examples of studies, and those data sets will be curated so that you can easily find the metadata and the different types of data that are useful in the analysis. So we will learn, for example, about the Cancer Genome Atlas and the data that could be found there, learning to navigate the Genome Data Commons portal, which is what you see right here, and how to find and prepare a multi-omics data set with all the needed clinical information about the samples. We will also talk about several curated examples that we have prepared so that we can learn about the utility of bioinformatics in precision oncology research. For example, we'll talk about a data set from the publication Modeling Precision Treatment of Breast Cancer, which was featured in Genome Biology in 2013. The publication is interesting because the authors assembled the data set of about 50 different breast cancer cell lines that represent the whole spectrum of breast cancer variation. The cell lines have genomic, transcriptomic, and proteomic data, as well as an efficacy measurement of commonly used and experimental cancer drugs. So we'll talk about some of the methods that they have utilized in this publication, and we'll see how we can run a similar analysis and the types of outputs that we get and what they mean for us to be able to select and understand the function of different treatments that have been described in this publication. We'll also analyze data from liver hepatocellular carcinoma, LIHC, which is a project on the Cancer Genome Atlas. And what we'll see is how we can find the various clinical data that is available on the patients that are described in this data, and how we can learn to use the metadata available in the clinical data sets to study the gene and microRNA expression related to these different types of patients. The different types of phenotypic data that we will see includes serology, pathologic diagnosis, BMI, obesity, and other risk factors. For example, infection with different hepatitis C and B, as well as alcohol consumption. We'll also learn how to find the data in publications associated with these studies and find the supplementary data sets that will help clarify what was done, how the patients were recruited, and what we can find about the different patients and the types of data that were collected from them. At the end of the program, participants will receive a certificate of participation if you have participated in the online sessions and have completed several different assignments that will be discussed throughout the sessions. We will also provide hands-on analysis practice. During those practical sessions, you'll be able to ask any questions and discover what went wrong in an analysis that didn't work. You will also gain access to one-on-one Q&A and assistance with any challenges or project preparation that you are considering for your own research or for your own study. You also get an account on the tBioInfo platform, which I will talk about at the end, that allows you to process efficiently large data sets and find all of the tools necessary for processing, analysis, machine learning, annotation, visualization of the data that we will discuss. So for the duration of the program, you have your account, and you can use that account with 5 terabytes of data storage, which allows you to process large data sets and get them in a form and shape that you can utilize either in a publication or a presentation. We'll talk about that as well. So again, to register, please follow this link. This link is available on chat. It's edu.tbioinfo.com, Precision Oncology 2020. And I want to just mention again that the program will start March 9th, 2020, and will last up until May 16th, 2020. We will have bi-weekly meetings every Monday and Thursday, and you can learn about the schedule and all of the topics that we discussed here on the link that we shared. Does anyone have any questions about this first program, Bioinformatics for Precision Oncology? Okay, if there are no other questions, again, we will share the recording and you can learn on the link that we shared with you, other topics and information. And if you have any questions, you can just respond to one of those emails. So the second program that we want to talk about today is the introductory program that we are launching for the first time for those of you in Africa, especially those of you in Nigeria. This program will be much shorter and will offer an easy introduction to the big data and life sciences challenges that are becoming more and more relevant, specifically in Nigeria and throughout the continent. And so to learn more about this program and start the registration process, you can also visit this link, edu.tbioinfo.com, OmicsLogic Nigeria 2020. It's the first program of that kind. Bipsha, if you could please share that link in chat as well. Thank you. So you can join this program. You can see that it's much shorter. It's only 15 days long, and it's really an introductory program. And throughout that program, we will also discuss a number of topics that we will continue to expand upon in future programs focused on that same audience. So the program is a 15-day online program that was developed by Pine Biotech together with several academic institutions and companies in Nigeria. In this program, participants will learn about the impact of genomic revolution, next generation sequencing, and computational technologies in every area of life sciences that include basic research, biomedical research, biotechnology, and agrobiology. We will learn how to find, understand, and then analyze data using high-performance computing as well as user-friendly tools for biologists. Sessions will be held online as well as on-site at the Biologic Support Services Limited in Lagos, Nigeria. The program is designed for students and faculty from life sciences backgrounds. In this program, we will speak about big data and how it has transformed the life sciences, as well as introduced both challenges and opportunities into every aspect of research, biomedical discovery, biotechnology, and agriculture. The program will be held online with 1-hour online sessions, as well as self-paced materials that are available for access online. So as you know, the omics technologies are becoming cheaper and are now used to explore the roles, relationships, and actions of the various types of molecules that make up the cells of an organism. Many types of omics data can be generated using next-generation sequencing, or what is also known as high-throughput sequencing. The data can show detailed information about genomic variants, epigenomic regulation, as well as gene and isoform expression. So we'll talk briefly about these different data types, and we'll discuss how these data types are used in different areas of research that have been mentioned. We will start the program with an overview of different types of omics data and the significance of the technologies that are being improved to generate this kind of data. We'll speak about next generation sequencing or high throughput sequencing and how new computational methods have been developed to analyze and process this data, as well as what are the challenges to interpret the data from the molecular level information that we have from every cell in the sample. So we'll talk about the history of omics data, availability of omics data, and some of the challenges and opportunities that we have in life science research related to the bioinformatics approaches that were developed to analyze and interpret this data. We'll also take a look at several examples of projects and several examples of data types that could be found on public domain repositories and are ready for anyone to take and analyze. We'll learn about some of the machine learning techniques that are becoming more and more available and powerful to analyze big data sets, including clustering, dimensionality reduction, as well as classification, and the impact of feature selection or interpretation of conditions that are sampled from various data types. This program will be an excellent introduction to the resources, materials, and a vibrant online community that you can join to continue learning about bioinformatics. Specifically, you will get to experience bioinformatics for yourself and try the following. Find and understand next-generation sequencing data. What is the data set or a project about? What is the question that you can ask or you are trying to ask? And can this data answer a question that you are asking? You will also process data, perform quality control, map, align, and quantify and evaluate different pipeline outputs. You will also analyze data by learning about the different statistical approaches, as well as exploratory data analysis, and interpret the data by annotation using reference databases and pathway analysis. Finally, you will consider an independent project and learn about future OmicsLogic programs that will help you make further progress with the project, with the research, and incorporate some of these tools into your analysis workflow. So we'll also talk about the student and faculty resources that we are developing to make bioinformatics more accessible throughout Africa and the region. The program will be available online, or an important part of it is that we will also offer it at the Biologics Support Services in Lagos, Nigeria. You can learn more about the company and their facilities on this website. You can see, 1 second, I will paste the link. So this is the link to their website. Sorry. Okay. And Dr. Toye is here with us also. So if anyone has any questions about this program or about the facilities or how the program will be conducted, please feel free to ask those questions in the chat box. The program is now open for registration. And at the end of the program, again, we offer certificates of participation and completion for those that are completing and doing all of the requirements of the program. That includes the attendance and doing some of the practical analysis. So you can learn more about this program and start the registration process using this link that was already shared by Beepsha. It's edu.tbioinfo.com, OmicsLogic-Nigeria-2020. So if there are any questions about this program or about the topics or about the participants, how this is going to happen, please let us know now. We're happy to answer any of your questions. Yes, I see that Redmi, you have a question. Okay, if you, there is an option to raise your hand, and then for Redmi, if you have a question, I'm happy to answer that. Okay. Okay, so if there are no questions about this, I am going to continue speaking about the next program that we're excited to be launching very soon. And this next program we will launch in May will be Bioinformatics for Infectious Diseases. In this program, we will learn about viruses and infectious diseases from viral and bacterial infections. We'll talk about zoonotic transfer of infectious diseases, viral infections, and bacterial infections. We will learn about analysis to that kind of data, including detection of viral genomes and next-generation sequencing data, vaccine design, viral strains and haplotypes, antiviral drugs and antibiotics, as well as study of epidemiological data and drug resistance. I'm sorry, I'm going to pause here because there's a couple of questions about the program in Nigeria. The question is the program in Nigeria restricted to only there, or can other individuals participate from other countries? Yes, it's in Nigeria on site at the Biologics Support Services, but online anyone can join. We did design the program specifically for people in the African continent. We have a foundation that has supported much lower rates for people to join that program for a very low and affordable rate. So if you would like to join that program, obviously anyone can join. This is an introductory program, and we would be happy to discuss with you access online for all of the sessions. Yes, thank you, Joe, for adding that about the biologic support services. Another question here, we will also get to learn about statistical methods that can be used to analyze sequence data. So there are multiple types of methods that we will discuss, starting from processing the data. So processing the data typically means that the data needs to be cleaned, and some of the cleaned up data has to be mapped on a reference genome. There are a lot of statistical methods that are involved in that process, but primarily we will discuss the statistical methods that are used for what's called downstream analysis, when we have to compare different groups of data, when we have to compare different signals and extract the signal from noise. And we will also talk about different machine learning methods that actually use computational automation for the different statistical principles that help us detect patterns in data that are typically not as obvious and not so easy to analyze with conventional statistical methods. So we will talk about those in different programs, right? An application to different types of data sets. The oncology will be focused on oncology data sets. This program, Bioinformatics for Infectious Diseases, will be data related to bacterial and viral infections. I hope that I answered your question there. Okay, so going back to Bioinformatics for Infectious Diseases, the program will cover different types of diseases, different types of analysis approaches, as well as provide you with some curated projects and data sets. And so we'll talk about viruses like Ebola, Zika, and dengue that have recently, we've seen large epidemic outbreaks. We'll also talk about coronaviruses like SARS, MERS, and the novel coronavirus in Wuhan, China, as well as some of the bacterial chronic diseases that have been around for a long time, but are also a concern, such as tuberculosis and leprosy. So we will start registrations for the program in March and plan to conduct the program starting around May 2020. We will primarily focus on analysis of viral genomes, and I'll give a couple of examples here in the next few slides. So with the recent epidemics becoming much more visible, dangerous, and unpredictable, there's really an urgency to learn about the different data and how to work with that data, and how can you use that data to understand the epidemiological spread of these diseases, as well as the underlying mechanisms that the viruses use to adapt to new circumstances, to new going from animals to humans, and others. And so we'll have a chance to kind of look at some of these infectious disease outbreaks that have been really impacting in the last couple of decades, have been impacting large numbers of people. For example, we'll talk about the novel coronavirus outbreak and how we can study the genomes from SARS, MERS, and the novel coronavirus outbreak in Wuhan, China, We'll actually talk about what data is available and how to find that data and what does this data mean. We will also learn about phylogenetic analysis and how it can be used to study viral lineages associated with our origin of the infection, time when that infection happened, and even the location. How can we find those associations? We will also look at some curated data sets that you can play around with. And the key in these curated data sets is that we actually break down some of the information, where you can find that information, how can you utilize that information, how can you actually download the sequences of those strains and other type of data that is available. So I think this is very important as you learn about these outbreaks, as the data becomes rapidly accessible. What can you do with it? Where can you actually find what you need to find to make a specific process clear to you from the data-driven research side of things? We'll also discuss different viral proteins, right? So we have the genome sequences. Those sequences could be found, but then we'll talk about how do we interpret the changes in the viral genomes to understand how those map onto the protein structures, and what is the functionality, and what are some of the insights that you can gain from comparing the impact of different mutations on the structural variants, and how we can also understand the differences between how those strains develop in animals as opposed to, for example, humans. So we'll have some data sets, and we'll have some papers and publications that we will discuss and do some of the analysis together to understand what does that really mean. We'll also talk about, for example, a data set taken from the Ebola epidemic in 2013, 2016. That epidemic has also yielded a lot of data sets. There's a question here for the infectious diseases training, particularly the viral infections. Will diseases like Lassa fever be considered? So again, we're right now in preparation for this program based on some of the research projects that we are doing that we are working on. And so we have an opportunity right now to consider a number of different viral genomes. And our purpose here is to really introduce the bioinformatics component of that. So once you understand how that works for the examples that we have prepared, for Ebola, for SARS, MERS, and other coronaviruses, as well as dengue and other viruses, I think you can kind of extrapolate how the same methods apply to any disease. We are not maybe ready to speak about the Lassa fever right now, but in the context of the program, we would be happy to help and support you if you're interested in that virus, so that you can start working on that project throughout the program. So as you remember from the Ebola outbreak, there's also a major component of understanding the different viral proteins, the changes in the viral proteins. How do we understand the relationships between the different strains and the different haplotypes of the virus, as well as how can we understand what mutations are important. And so in the context of that data set, we will really look at the spread of the infection and really monitor how the infection spread and how the viruses are able to replicate and change as a result of this spread throughout a large region and over a period of almost 2 years. So we will discuss how the different process could be then studied using data that is available in the public domain from the different patients that were sampled, and a lot of the studies that were done afterwards in cell culture as well. So we'll really break down the data set, help you find the necessary annotation for the different samples, and provide you with several examples that we will actually work on together throughout the program. We will also talk about how the studies and the bioinformatics approaches were actually designed to study the increased infectivity, the different mutations, And also we'll talk about how that has led to the development of a vaccine that is now available. 1 second, there is also another question about the precision oncology program. Will we be able to work on data sets specifically for metastasis, such as topics like breast cancer metastasis to brain and bone? We will also be able to learn about the drug targets and therapy targets for cancer metastasis. Okay, so excellent question. Thank you, Chandrani. We do have a project data set on brain cancer, lung cancer, and breast cancer. And we will discuss in detail the process of metastasis and how does it start and how to detect it, and what other methods, like, for example, exosome analysis of exosomic DNA and RNA, is available for us to study early detection of metastasis. So we will talk about that. I think if your question is specifically, will we focus on one type of cancer, like the breast cancer, and how it metastasizes, we will not necessarily have an in-depth discussion just focused on one cancer, but we will have different examples. And I think the objective of these programs is to provide you with tools and information that gives you enough understanding and practical skills to be able to develop a project of your own. And then as you work on a project of your own, we will be here to support your project and help you make progress enough in the project to be able to then present your work to your peers as well as to experts and get some feedback. So a lot of the topics that you're mentioning will be touched upon. Whether we will do a comprehensive overview is really remaining up to you to decide that will be your project. And so we will continue to work on that at the later stages of the program. Let me know if that makes sense. Okay, so we will again, in the infectious diseases, focus on Ebola a little bit and talk about the different mutations that were found and reported on. And we will also prepare some curated data sets that you can learn from, working on them independently and seeing how the different data analysis tools really are used by researchers when they study these kinds of epidemiological data sets. And obviously, one of the outcomes of that data being available is the design and the development of different vaccine approaches, as well as different antiviral approaches, antiviral drug design. And we'll talk about some of those examples as well, and how that is being successful as a result of different bioinformatics efforts and the availability of this kind of data. Now, do you have any questions specifically about the program for infectious diseases? And again, you can put your questions in the chat box. Okay. So the next portion of the presentation, okay, registration, sorry, there's a question. How can I find info about registration? If you mean the registration for the infectious disease program, in March, we will announce the registration details. So please make sure that the emails that we send from Pine Biotech, you will see that we send regular monthly emails. So either you can reach out to us through this email. Sorry, that was meant. Yes, thank you, Bipsha. For everyone, you can reach out to us here. And with questions like, what is the pricing? What is the availability? How do you access? Any questions, please feel free to reach out to that email. But specifically about the registration for the infectious disease program, we will be posting that announcement on our website. But also, obviously, we will be able to send you an email with that information. So if you leave your information with us, your email address or contact information, we will reach out to you once we have all of the details for this last program. Okay, so the other thing that I wanted to talk about is really the significance of knowing how to utilize bioinformatics for every aspect of data analysis, from experiment design, where sequencing methodologies and analytical methods are critical because of the data generation process, to bioinformaticians that ultimately are facing new challenges every day with new methods and new questions from the biologists, and all the way to interpretation and utilization of these data sets and insights from the analysis itself. And so here, we are really trying to offer affordable and easily accessible bioinformatics training that is focused on biology as a data science, which we believe is really what it is becoming. And to address this challenge, we have designed a number of materials that we have been developing together with different research institutions, as well as testing it on different audiences, from clinicians to biomedical researchers to students, all the way from high school, undergraduate, and graduate levels. So we're really trying to prepare structured materials for anyone interested in learning about bioinformatics for different types of omics data. And these include Introduction to Bioinformatics, analysis of genomic data, transcriptomic data, epigenomic data, metagenomic data, as well as a series of courses on machine learning and data science. And so these materials are all available on our main website. The main website is this one. And you can go on this website and see under courses how these materials are organized. Now, the unique aspect of these materials that we are utilizing in a lot of the programs that we have discussed up until now is that these programs are available in a personalized way. So you create an account. That account gives you access to specific types of courses or even series of courses. And you can also track your progress and always go back to the place where you left off, as well as measure your participation, your progress, see it by analyzing the different data sets and getting credit for all of the activity that you do. So these materials, we also ask our participants to provide us with some feedback. And so we are really excited that we are getting a lot of feedback, positive feedback from the people that take these courses. Now, you have to understand that the courses are designed for different levels of students. Some students are very advanced with a focus on their expertise in life sciences, or some people that are participating in a school curriculum or a course at their university. And so really the different levels, if you want to talk to someone and understand what is an appropriate level for you and what is the level of difficulty or technical skills that are required in a specific program, please reach out to us. We're happy to discuss with you what you can do to find a starting point for wherever you are in your bioinformatics journey. And so we've seen a lot of good feedback about the online materials. And so I want to talk a little bit about how these materials are organized and what they can give you. So like I said, anyone that joins the platform is given an account. Some of the courses are completely free. Some of the courses have minimal payments associated with them. But basically, as a part of that training, you belong to this community where you can see your own progress, you can see your activity, you can also join a program that is organized, and there's going to be a group of people that are going through the same journey. And so some of the participants that perform exceptionally well are also featured. And you can see how your progress, your work, your contribution actually helps you get visibility and get really recognition for the amount of work that you are doing. So you can find some of these top performers here on the leaderboard. And maybe, Bibsha, you can post a link to the leaderboard in the chat box as well. So you will see how you can navigate some of the top profiles and see what programs they were a part of and what was their journey. And we also highlight some of the participants in our blog to share with our community how these people joined the program and what have they gained from the different programs that we have done. Now, all of this information is on the main site, edu.t-bio.info, but we also have other sites that are specific to different regions. So if you have any questions about that, you can also reach out to that. Now, the courses themselves are designed in a way that you can easily navigate the different sections of a course. You can see different key terms. You can also search for different topics and also download a PDF version of the material. If you're offline, you can still continue to learn. There's also a forum discussion where your questions are being answered, and you can ask any of the questions related to a particular course, topic, or lesson. And obviously, you can interact with our curriculum development team that is focused on improving the materials and making sure that the materials that we have are interactive, easily accessible, and help you learn some of these theoretical and practical skills. Now, also the learning is supported by our online bioinformatics research platform. This platform could be used for research using a research account, or it could be used for education, where you learn using the curated data sets that we provide, and your points are accumulated under your profile for each activity that you do. The platform is designed in such a way that you can find clear explanations of every step that you do with a lot of different varieties of methods that you can compare and contrast. And you can also learn without any coding, but it doesn't stop there. You can also take it further once you learn about the different types of methods, about the different analysis, about the different data sets, and about different questions that you might be able to ask of the data. Now, the platform, you can find more information about the platform and the different types of analyses and the different types of data sets that could be analyzed with the data. Bibsha, again, if you could put this link in the chat box. In this platform, I'll just give a little overview. You will find materials on how to use the different sections. For example, you can see here transcriptomic data, including single cell analysis of transcriptomics from single cell, ChIP-seq and bisulfite-seq, analysis of mutation variant data, copy number variation, analysis of non-coding regions of the DNA, as well as the repeats that are found in the DNA, microRNA analysis, analysis of microbiome, and 16S sequencing from metagenomic data, mass spec, structural biology analysis of structures. And you will also find some focused types of sections, for example, for virology and for data association and integration, as well as machine learning, data mining, and some utilities. So it's a really broad platform that allows you to learn the logic of the analysis and does not require any coding for you to get started. Now, once you start and you learn about these different methods, we also provide with some coursework as well as examples that you can start translating those methods into your own pipelines. For example, we discuss different packages in R, how to start analyzing and exploring your data in the RStudio environment, and many other options like that as well. So make sure that you check out the platform. Those of you interested in the platform itself for research, we have that possible. We also have different support services that you could request. And so you can learn about some of the pricing and some of the options for those support services on this link as well. Okay, so that's the platform. The platform is intuitive and really does not require a lot of guidance. And so anyone that is just learning bioinformatics is able to use and learn as you use how to apply these different methods to the different data sets. So here you saw a quick example of building a complicated data set. And you can also see that a lot of the outputs are going to be visually easy to interpret and work with as you understand what type of data you are working with. And we obviously provide additional information in some of the courses that we have designed, and some of the programs provide you with guided, assisted kind of version of the same tools and data sets. When we do these projects, I mentioned some of the projects today from oncology, from viral and bacterial infections. We also have many others. And what we do basically is we extract data from top publications in different areas. Those areas include oncology, neuroscience, infectious diseases, and others. And what we do is we extract the methods from that paper and kind of replicate the analysis that the authors did. But once you understand the data and the methods that are described, what you can do is you can actually combine the two, learn how to combine the two into a novel and independent project of your own. And so those are really the objectives of these OmicsLogic programs, is to help you design a project of your own using this kind of guidance. So we have many different projects available. For example, epigenomic analysis of primary human T-cell differentiation and asthma susceptibility, modeling precision treatment of breast cancer using multiple types of cell lines and therapeutic agents, viral mutation landscape study using ultra-deep Seq sequencing of polioviruses, Grassmannia clavigera adaptation and toxic resistance and pine pathogenesis, pathogens, analysis of the changing immune response in cancer, microbiome biomarkers from the American Gut Microbiome Project, whole transcriptome profiling of cancer tumors and mouse PDX models, deregulation of subcellular networks in cancer patients, insect cell lines biologics production, Spotoptera frugiperida, and rhabdovirus contamination, how to find and analyze viral sequences in this kind of data, crop drought stress and resistance using the potato cultivar, different cultivars from the potato species, as well as complex gene regulation, neurodegenerative diseases, Ebola, the deadly mutations, and finally, your project, right? So after learning from these examples, the examples that were taken from curated publications with curated data sets and prepared for you, you get to apply the same skills and the same understanding from these programs to your own data set and your own project. So all of these are taken from publications, like I mentioned, and transformed into guided tutorials that you can either take online and follow yourself, or join one of the programs where these project examples are used in the training sessions that we conduct. Now, at the end of these programs, oftentimes the students and the faculty that have participated in previous programs, they help us prepare the projects because they have analyzed the data, explained all of the data, and then have gotten feedback from their peers and from others on the types of projects that they have done. So that's really our objectives. And next, I want to briefly explain about a couple of opportunities for any one of you that is joining one of our programs to do that, to present your project to others. So the first one is a project that we are doing together with University of Engineering and Management. This is a Bio-Olympiad. And Beepsha, if you could please paste the link to that Bio-Olympiad site. This is a program that it's actually an online competition that is organized by the University of Engineering and Management in Kolkata in India, in association with Pine Biotech and the Society for Makers, Artists, Researchers, and Technologists called SMART. This online event is organized to develop interest and competitiveness among students with an interest in life sciences. And our goal for the participants entering into this Bio-Olympiad will be an inspiration among the young learners in a variety of applied biological sciences. Now, this specific program is only for students that want to do problem solving in applied biology within the age group of 16 to 24 years old. The contest is open to participants from across the globe. However, they need to abide by the rules and regulations of the contest. There is a prize for people that will participate in this contest. The first 3 rank holders will win a prize worth $300 US dollars in total and merit certificate. In addition, the top 10 people will get merit certificates according to their score. There are a few rules and regulations, and it's free to participate. And the actual competition will be online, but you have to actually register to participate in this competition. So please follow that link that Vivsha posted, and you will get more information about that. Okay. Another opportunity for those of you joining the programs is an upcoming program conference on computational biology and bioinformatics that will take place in April 3rd and 4th, 2020 at the Louisiana State University in Baton Rouge, Louisiana. Those of you that are participating in any of the programs that we have a program with the Louisiana Biomedical Research Network, and we have programs with different universities like the Amity University or University Institute of Genetic Engineering and others. You can reach out to us about participation in this conference. We will actually help you prepare your project poster if you are in any one of our programs and would like to present your poster during the poster session at this conference, which of course you can put on your resume and you will get feedback from experts. And there are a lot of high profile speakers at this conference as well. If you would like to join, different topics will be discussed from cancer informatics and immunotherapy, microbiome and metagenomics, proteomics, cloud computing, evolutionary genomics and phylogenetics that we talked about today, virology and infectious diseases. And so really a lot of the programs that we have been discussed will be also covered by experts in this conference. And you can either join in person or reach out to us to get more information after the conference is over. Now, previous participants of our programs have been able to participate in this program, so you can see some of the people presenting their posters from different universities, and we also will have a workshop during that session. You can submit abstracts and projects done already if you have not published them, obviously. I am going to paste here a link for anyone that is interested. You will see more details about the conference itself right here, and also the registration information that you can join. And some of the speakers are described right here as well. So please take a moment to take a look at this. And obviously, if you have any questions, I would be happy to answer those as well. I will finish with, okay, so there's another question. The question is whether you can submit abstracts on projects that are being done with publicly available data sets. So I will mention that, yes, many of the projects that people undertake, many of the poster sessions that people do utilize online data, to perform new analysis, right? So one of the papers actually that we will discuss in the oncology program are papers that, utilize existing cell lines, but test different new methods and develop new results and insights from those data sets and from the analysis that they perform. So definitely you can use existing databases, you can use existing data sets. You probably need to develop some novel aspect of the analysis or interpretation and do some literature review that indeed you are providing some value to the research community. And that's why that abstract submission is going to be evaluated by a panel of experts that have expertise in both bioinformatics, but also some of these domains that we talked about, the cancer, infectious diseases, immunology, and others. Okay, so again, if you have any other questions, I would be happy to answer your questions. We have also online here with us some of our trainers and some of the other organizers of the individual programs that I mentioned here. So if you have any questions for them, we can answer those as well. Okay, well, I hope that we were able to answer most of your questions. If you have any other ones, feel free to reach out to us by email. You will also notice a chat box on our websites where you can ask questions as well. Make sure that you check those out. And also, we have here Bipsha and Mohit from our program in India. If you have any questions specifically about Indian locations or dates, we have some upcoming workshops and opportunities to learn there as well. You can reach out to us, to them there. We also have several US programs right now at LSU, like I mentioned, and throughout the state of Louisiana, as well as at Georgetown University and others. Okay, there's a question here. How long will the certificate be valid for? So the certificate is issued for a year. We actually specify the date on the certificate. So 12 months from that, the certificate is valid because we expect to cover a lot of the methods that are relevant, but the field is dynamic. The field of bioinformatics is dynamic. And so we encourage people to continue and learn after they get a certificate from a program. However, this certificate is really going to demonstrate that you have participated in a program that has been evaluated rigorously by a variety of academic and industrial institutions. It is in collaboration with the Tauber Bioinformatics Research Center from University of Haifa in Israel, the people that have developed and continue to develop the T-Bioinfo platform. It's also in collaboration with a number of other organizations that we mentioned on the certificate. So the certificate is going to be good for the next foreseeable future, but we do encourage you to update your training at least once a year and participate in programs that provide you with updates to the different technologies and methods that are emerging in these bioinformatics markets. Okay, for those of you that maybe just recently joined, we are just wrapping up here. We will send the recording of this webinar with all of the commonly and frequently asked questions in the follow-up email. Please look out for that email. We've covered a lot. I'm just going to, again, review some of the upcoming programs just so that those of you that are interested, you can see what we are planning in the near future. We have a program upcoming for bioinformatics for precision oncology, a program that is an introduction to big data in life sciences, as well as bioinformatics for infectious diseases. And we discussed today a lot of the online materials and resources that you can utilize in your research, as well as to learn about bioinformatics, starting from basic and beginner level all the way to advanced research level. Thank you again for joining this webinar. If you have any other questions, please make sure to reach out to us and look out for those upcoming emails where we will be sending you more information about these programs and online materials. Thank you again, and I look forward to seeing you in one of these upcoming programs.